I'm Susan Amara. I'm the Scientific Director of the National Institute of Mental Health. And I'd like to welcome everyone to the NIH Director's Wednesday afternoon lecture series. And this is the at-home edition. Many of us are still at home. The WALS will be held remote only in this way until through December. And so we're not sure yet what will happen in January, but at least through December, the WALS will take place remotely. I have just a few announcements that they've asked me to make before we get started. Today's not so secret code for CME is 31278. That's 31278. Also, if you have questions for the talk, please submit them via the videocast website and look for the words that say send live feedback, and it's below the video display on the web page that you're watching the lecture. The questions will ultimately get to me, and I'll ask them to Mark. I'll pose them to Mark, and towards the end of the lecture, we'll answer the questions. You can submit questions anytime during the lecture, and I really encourage you to ask Mark hard questions. Mark has never failed to ask me very challenging questions when he's seen my talks, so feel free to fire away. And now I'd just like to introduce the speaker, Mark Nelson. He's a University Distinguished Professor and Chair of the Department of Pharmacology at the University of Vermont. In my mind, Mark Nelson is a consummate physiologist, biophysicist, and his lab has really sought over many years using wonderfully elegant techniques that you'll see today to understand the control of smooth muscle and endothelial cell function by ion channels, calcium signal. And his work has really emphasized this cerebral circulation. And he's been interested in diseases that relate to small vessel disease, or SVD, which he'll probably mention during the course of his talk. Dr. Nelson received his BA in mathematics and biology from Tufts University and his PhD in neural science from Washington University in St. Louis. After a number of fellowships in the 80s, including one with Professor Peter Lauger at the University of Constance in Germany, he joined the faculty at the University of Vermont, and he rose to the level of chair of the department in a relatively short time, in achieving that in 1996. And for the past decade, he has held professorships both at University of Manchester and University of Oxford in England, joint professorships. So many of us think of the brain as a complex network of neural connections that enable communication across distances and enable sensory inputs and all the things. But rarely do we think deeply about what supports, about all the cells and networks that actually support those cells. The Nelson Lab has illuminated the mechanisms by which brain capillaries act as neural activity sensing networks by initiating and transmitting electrical signals through interconnected endothelial cells that are really comprising the lining of capillaries. Using a mouse model of monogenic small vessel disease, they've discovered that early defects that result in a loss of electrical signaling and impaired delivery of blood to active neurons. These defects often involve changes in extracellular matrix composition. His research interests have included elucidating the mechanisms by which cerebral blood flow is controlled to meet all the demands that neurons and chemical communication really demand within the brain. And he's also studied how these mechanisms are disrupted in small vessel disease, which are a major cause of the deficits in stroke and dementia. He's unraveled many of the signaling mechanisms, including an interesting discovery of local calcium signaling, which he has referred to as sparks, which actually counterintuitively oppose vasoconstriction. In 2019, Dr. Nelson was elected to the National Academy of Sciences. And he also recently received the NIH Outstanding Investigator Award, an extramural award, the R35, which provides his lab with long-term funding. I can say I think he's been funded by at least 5 different institutes that I could count. So his work really impinges on work that occur in a variety of different fields. And in that way, I think is very multidisciplinary and has implications not just for the brain, but for anywhere where smooth muscle regulates vascular function. The title of Dr. Nelson's talk is Translating Thought into Blood Flow in the Brain, Capillaries as Sensors of Neural Activity. Mark, thanks so much for taking the time to come and talk to us. I'm really delighted and honored that you could come. So thank you.
I'd like to thank Susan and NIH for inviting me. It's certainly a thrill. It was a tough trip down to NIH, but so far we're doing pretty well. This first cover slide is lifted from the University of Vermont, but it's what Vermont looked like maybe a week or 2 ago. In the middle, that shows in vivo image of a volume of a mouse brain, and little green flashes are flashes of calcium in the vascular endothelium, just sort of for a little intro bling. I'm trying to go to the next slide here. What's going on here? All right, there we go. First of all, I'd like to give you a little bit of background and thank the NIH, Susan, for the support. I listed many supporters from the NIH for our lab, including Heart, Lung, and Blood Institute, which I have an R35, NINDS, and NIA, which I have the R01, NIDDK, which I had an R37, and Mary Cushman and I at the University of Vermont just got a COBRA grant from General Medicine. Plus, we've had other international funding groups, which I will talk about later. This is the University of Vermont. Here's the Leukoencephalopathy College of Medicine. There's our lab. This is my office where I'm sitting right now. And this was probably a couple weeks ago, several years ago. This is Lake Champlain. The Leukoencephalopathy College of Medicine at the University of Vermont is the 7th oldest medical school in the country. We have a relatively new dean, Rick Page, which we're delighted about. And we just set record funding of $105 million for the College of Medicine this year, and record funding for the university. So even though we're 500 miles northeast of you, we're doing pretty well. This over here is the hospital. So let me tell you a little bit about what we're working on. Hopefully you'll find it fun. I invite you all out for a drink later on, and just send me the bill. I'll pass it on to the dean. Just kidding. So our goal is to understand the control of blood flow in the brain, as Susan said, and how it degrades with disease and age, and hopefully try to find new ways to slow this down or even correct it. A little bit of background for everyone. The brain represents 2% of the body mass, consumes 20% of the body's energy, for most people, that is. Neurons are a just-in-time machine. They have limited energy reserves and depend on on-demand delivery of blood, known as functional hyperemia. Function means obviously function. Hyperemia, hyper, increase in blood. So we'll be talking about functional hyperemia, this on-demand delivery of blood. Dysfunction of the small blood vessels within the brain, mixed with Alzheimer's pathology, is the major driver of dementia. And the financial and societal impact of dementia, as everyone knows, is staggering and increases as the population ages. On the left is just sort of a simulation of neural activity in the brain. But just think about that. Every time part of the brain is active, blood is delivered to that part precisely and rapidly. And this happens in us every moment of every day, while waking, asleep, year in and year out. And just as the heart beats reproducibly for year in and year out, this works like that. And we don't appreciate it until it's not working. I want to give you some insights into this, hopefully. Just for a little more background, these are stats from the Vermont Department of Health. And what it shows is that, that the population in Vermont is aging here 70 to 74, 75 to 79. And that's the biggest increase in the population in Vermont. So the population in Vermont's aging. And statistic, 1 out of 7 Americans over 70 has dementia. So this is a huge burden as the population ages, and particularly in Vermont, where we see the younger population are declining, which has been of serious concern for a number of years here. So it's just a little background. This is a vascular cast of a human brain. This shows the vessels, the surface arteries that then penetrate as arterioles and penetrate into the parenchyma of the brain, and a vast network of capillaries. Now, what you first step in the process of the talk, imagine that all these blood vessels are all interconnected, and the cells that line all blood vessels are called endothelial cells, and capillaries are composed solely of endothelial cells. And all these endothelial cells are electrically coupled. So if you stimulate them and they change the voltage, the current will move from one cell to another. So you can view this as really an electrical circuit within the brain and communicating to one another to help support the activity of neurons. A few factoids useful for Friday night, chatter. The human brain has about 1,000 miles of blood vessels. The vast majority are capillaries. The smallest blood vessel, the capillary, touches every nerve cell in the brain. And just for perspective, a capillary's width is about 1/20 the width of a human hair, which is about, in this case, maybe 10 to 20 microns. And also another little bit of perspective, red blood cells and capillaries, the size of them are really no different in a rodent than they are in a human. So when we get down to these dimensions, which is really very, very parallel to humans. What I'd like to do is provide some evidence or show you some evidence that capillaries sense neural activity to send signals electrically upstream to signal for the need for blood. A little bit more about background. This shows you a little experiment from someone else looking at the somatosensory, the sensory cortex, which responds to whisker stimulation in a rodent. This shows the neural activity here. The local blood flow into the somatosensory cortex within seconds rises to deliver blood to meet the demands of these active neurons. This phenomenon, functional hyperemia, has been known since the 19th century, and this shows one of the prominent papers from Roy and Sherrington. They're pictured here. The brain possesses intrinsic mechanisms by which its vascular supply can be varied locally and corresponds to local variations in functional activity. So the central question, which I'll provide some evidence for at least one mechanism, is how is neural activity translated to increased local blood flow? As many of you are aware, and many people use the technique, functional MRI, brain activity is monitored by changes in blood flow or deoxygenated blood, shown here as an example here. And so interpretation of these changes presupposes that there's a one-to-one correspondence between demand for activity of neurons, say in this spot, and the demand for blood flow. There are also examples where blood flow might go up somewhere else that's not related to the local activity of the neurons. So it's pretty complex, but certainly it's a non-invasive way to look at changes in blood flow and neural activity. So I'm going to give you just an overview here of what we think is a major mechanism that accomplishes this rapid local delivery of blood. So when a neuron is active, fires action potentials, electrical activity causes a release of potassium from the neuron during every action potential to repolarize. Well, we believe that this causes a local increase of potassium around the capillary endothelial cells, and there's a potassium channel called the inward-rectifier rectifier 2 channel members. They're called strong inward rectifiers. They are exquisite sensors of external potassium. And if somebody's interested, go through the mechanism. They turn on this inward rectifier potassium channel. It causes a hyperpolarization of the endothelial cell. These channels are also turned on by hyperpolarization, and then the signal is transmitted through gap junctions upstream to the penetrating arteriole, through gap junctions to the smooth muscle cells to cause hyperrelaxation of the smooth muscle cell. And we've estimated this signal travels in an excess of 2 millimeters a second and has all the properties you need for a rapid upstream dilation of the arterioles to deliver blood downstream. So I'll show you some examples starting at, asking the question, is the sensor here, the ion channel? Can we show this property in an intact ex vivo prep and then ultimately in vivo? And then we'll talk about some of our recent work on what goes wrong in small vessel disease of the brain. So interestingly enough, when we started doing this a few years ago, it was that we asked a very simple question. What are the ion channels? What are the electrical properties of capillary endothelial cells in the brain? And this is largely unknown, certainly from freshly isolated cells. So we set up a procedure to freshly isolate endothelial cells from brain capillaries, then using the patch clamp technique, which basically involves measuring current across the whole cell of a single isolated capillary endothelial cell, we can measure membrane currents. And I know everybody loves this inward rectifier potassium channel, but I'll show you a few currents anyway. So that it's characterized by letting potassium go easier into the cell than out. So this downward current is an increase in current to changing the voltage. And one of the features of the channels is rapidly and potently blocked by barium. And barium ions plug the pore. So it's a useful tool in combination with genetics to try to look at the function, the role and function of these ion channels. So barium, you'll see barium used throughout. It's fast. It's a potent blocker of this family of ion channels. We've looked at all the other ion channels in endothelial cells and smooth muscle cells, and the concentrations we use, barium has no effect. And we use this in combination with endothelial-specific knockouts of the inward rectifier potassium channel as well. So we wanted to go to the next level and look at how capillaries affect arteriolar function in an isolated preparation. And Fabrice Dabertron, shown here, very dapper, developed this preparation when he was here. Now he's a faculty member at the University of Colorado. It involves taking arterioles from within the brain, cannulating them, and leaving the capillaries attached, as you can see here in green. And you can see one branch, another branch, another branch over here, and then stimulating the ends of the capillaries and looking at the arteriole up here that's cannulated. And this shows, the next thing, hopefully it works, the slide, shows an example of the functional response. Yep, it's working. Here's the capillaries in what we call the Fabrice prep. And you can see this is the upstream arteriole. We spritz on potassium here at 10 millimolar. Physiological potassium in the brain is 3 millimolar. And we measure the blood vessel dilation or diameter up here. You can see this roll around again. And the diameter of this arteriole, it's pressurized at 40 millimeters of mercury, so it's partially constricted, is 15 microns. So you can imagine the challenge, which Fabrice can do this in his sleep, of cannulating a 20 micron diameter blood vessel. This dilation to potassium is rapid, it's blocked by barium, and doesn't occur in the endothelial-specific knockout of the inward rectifier 2.1 channel. I should also mention that I wanted to name the preparation after Fabrice, La Fabrice, but the journal thought that wasn't probably an appropriate title for the preparation. The next step, we go in vivo to test it using a multiphoton microscopy, which we can then look at the vasculature up to 1,000 microns, usually about 300 microns in the somatosensory cortex by making a cranial window and then having the genetically encoded, depends on what we're doing, genetically encoded indicators or measuring red blood cell flux and velocity. And I'll show you this right here. So the green is fitzy-dextran, which we've injected IV. This is an artery up here on the surface of the mouse brain. This is in vivo. The mouse brain is fixed in place, and you can see all the blood vessels. See smaller ones here and here. Then we zoom in to a capillary. And note the green spot of the dye in the plasma. The black dots are single red blood cells that occlude the dye. And then, this, just for orientation's sake, a red blood cell in us or our mouse is about 6.5 microns in diameter. The capillary lumen is about 4 or 5 microns. So the red blood cells go single file through the capillaries. We can then scan the capillary. We can then count the number of red blood cells per second in vivo. And then we can look at the velocity by looking at the angle of this sweep as the line scan goes through. In doing that, we can also then place a second pipette filled with a red dye into the brain, onto a capillary, just downstream from an arteriole. And this shows the placement. You can notice in the bigger vessels, you just see these little rapid black streaks. Those are thousands of red blood cells going through, side by side. Okay, so we place the pipette, and within a few microns of a capillary, and then we puff for 300 milliseconds, slightly elevated potassium. And what we see is that the red blood cell flux, in this case, more than doubled. So the idea is that potassium comes out 10 millimolar ever so briefly, turns on this inward-rectifier potassium channel, hyperpolarizes, the current goes through the endothelial cells upstream and leads to a dilation to upstream arteriole. And for the neurobiologists out there, think of this as an inverted action potential. Instead of a depolarizing action potential, this is a hyperpolarizing response that can regenerate and travel rapidly upstream or maybe downstream to cause an increase in red blood cell flux. And this shows an example, baseline 10 millimolar potassium, and there's an increase. You can see that each streak is a red blood cell, and this is 1 second that you're shown here in the line scan. So in this case, the flux almost doubled with picospritzing 10 millimolar potassium for 200 milliseconds, 200 to 300 milliseconds. All these responses were blocked by barium, which blocks the inward rectifier, 2 family members, and they were also abrogated in the genetic knockout of the endothelial inward rectifier 2.1 channels. The next step is we then go, some messiahs on this work, we go in vivo to measure cortical blood flow in the somatosensory cortex in a volume of it, using laser Doppler fluorometry, which looks at the backscattering of light in response to the movement of red blood cells. So here, WS is whisker stimulation. Red blood cell flux goes up, increase in cerebral blood flow, and about 60% of this increase is blocked by barium. Then we went to the inward rectifier 2.1 knockout, only in the endothelial cells, and the response is reduced by about 50%, consistent with this mechanism, this rapid retrograde electrical signaling being responsible for about 50% to more of the increase in blood flow to whisker stimulation. So this sort of, again, same cartoon, the concept is neural activity releases potassium directly or indirectly through astrocytes. The local perivascular potassium goes up ever so slightly, turns on the inward rectifier potassium channel, sends a hyperpolarizing inverted action potential through gap junctions upstream to cause relaxation of the smooth muscle cells, increase in red blood cell flux. Barium blocks it, abrogated, blocked by knocking out this ion channel endothelial cells. And this shows us sort of another way of looking at it. And we view this system, this electrical system, and the capillaries as sensors of neural activity as a nervous system within a nervous system, sending signals against the retrograde against the blood flow to deliver blood precisely and rapidly on demand. So we called it the shadow nervous system because in a sense it's monitoring, shadowing the central nervous system. Well, at the same time, we started a program with Andrew Tell, supported by the Leduc Foundation, and we were awarded a transatlantic network of excellence on the pathogenesis of small vessel disease of the brain in 2012. And this is to really support North American and European collaboration. This shows Anne, Martin Dichons, one of our major collaborators, and the various groups that were in our network. And so we had clinical arms of the program, basic science ones, and molecular biology ones. And so this was a wonderful opportunity to really make the science go international. And toward the end of our Leduc network funding, which ended last year, Martin also applied to the European Union as there's Andrew Tell, me, Frank Ferrazzi, as PIs for, this is our Leduc network, but he applied for a grant from the European Union. Just to show you how international this is actually a picture of one of our Leduc meetings at the University of Manchester, where I have a collaboration, as Anne mentioned, with Adam Greenstein and a position there, just to show how important that international science is. So we're in one of the major research buildings at the University of Manchester. And the president of the University of Manchester, Dame Nancy Rothwell, came and gave us a talk. She's a stroke researcher as well. So that was a group picture. This is our Horizon 2020 group picture from last year. And there's Martin and Andrew Tell, and Nick and Masayo from the lab here, taking advantage of being in Paris in the summer. So this has been wonderful opportunities to really amplify our research and also to go to Europe. So I want to give you a little bit of background on small vessel disease of the brain. It's responsible for about 30% of the ischemic stroke, more than 90% of the spontaneous intracerebral hemorrhages, the most devastating subtype of stroke. It's a leading cause of age-related cognitive decline and disability. Major risk factors really are hypertension, age, I don't know. As I get older, I never call age a risk factor anymore, and some genetics. It progresses like many diseases for many years before becoming clinically symptomatic. There's really poor understanding of the mechanisms, and to date, there's been really no specific treatment. There are a number of genetic causes of small vessel disease, and this is how we got into it. And one of the ones we've been looking at is CADASIL, and other one, collagen 4A mutations. And so I'm going to just give you some more recent work on CADASIL. So CADASIL, cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy, is the most common hereditary form of small vessel disease. It's about 1 in 50,000 people have CADASIL. So we have patients at our hospital with CADASIL. It's autosomal dominant. It has all the clinical and MRI manifestations of sporadic small vessel disease. It's mutations in the Notch3 gene in the extracellular domain. And this is, Notch3 is only expressed in smooth muscle cells and pericytes in the brain. It leads to accumulation of deposits on the small vessels in the brain. And we use the mouse model as a window into this genetic model with the hopes of gaining insights to the more common sporadic forms of small vessel disease. This shows you a little bit about the natural history of cerebral vascular manifestations in CADASIL patients, starting in the late teens. Many of the patients, not all, have migraine with aura. They develop white matter hyperintensity, so these are lesions in the white matter where the axons are in the brain. Cognitive deficits start often in the 30s. Later on, motor disability. Many of the patients die by age 60 of stroke. But the main thing is that they also have compromised cerebral blood flow regulation, which is true of pretty much all vascular diseases of the brain. And they have these deposits that are quite characteristic. Well, Andrew and colleagues developed a mouse model over 10 years ago that has one of the human mutations of CADASIL engineered in, and it had all those the characteristic early stages of disease, including white matter lesions, decreased cerebral blood flow, which is the focus of our work, accumulations of the same deposits of proteins around the blood vessels, inward remodeling. This is the aspect I'd like to talk about, is what happens to blood flow control in the brain and particularly functional hyperemia. So what we found is that the whisker-stimulated increase in cerebral blood flow in the somatosensory cortex was reduced by about 50% in CADASIL. And this looked very much like our inward-rectifier 2.1 knockout. And to determine how much of this was the barium component, the inward-rectifier component, this is without barium put on the surface of the brain. This is with 100 micromole barium, and about 60% of the increase in blood flow to whisker stimulation is gone. And it's mostly what's lost in CADASIL is the barium-sensitive inward-rectifier component of functional hyperemia. I should also mention part of our Leduc network, Chabriot and colleagues showed that CADASIL patients also have a deficit in functional hyperemia, even at an early age. In this case, I looked at using functional imaging techniques in humans. So this shows sort of the thought here. K goes up, changes electrical properties, leads to smooth muscle relaxation. And this is crippled in this model of small vessel disease. We tested this in our ex vivo arterial capillary preparation by picospritzing potassium on the ends of the capillary here, and then directly on the arteriole. So pipette 2 is on the capillaries. And what you can see here is this is the diameter of the arteriole here, and when we picospritz potassium, the upward deflection is a dilation, direct stimulation of the arteriole, nice dilation. If you remember the movie before, when picospritzed on the capillaries, we got a really profound and rapid dilation. Now in this model of small vessel disease, no dilation, nothing. Then we do back the arteriole, works, capillaries don't work. The capillary stimulation, this whole mechanism is gone in CADASIL. So then Osama looked at the electrical properties of capillary endothelial cells from this CADASIL mouse model, and showed that the currents were reduced by about 50% in CADASIL, which based on direct measurements and computational modeling with our colleagues in Florida, that this is more than sufficient to cripple this electrical signal. Also, Osama found that the only defect in the ion channel was in the capillary endothelium and not in the arteriolar endothelium or the smooth muscle. So we were thinking, if you look at this, we're thinking, well, how could this be? You could have fewer channels, you could have them endocytosed, you could have them blocked, a whole variety of reasons. But one reason we jumped to, which because it's already been shown by Don Hilgeman and Rob McKinnon, is that the channel activity of this type of channel absolutely requires a phospholipid, phosphoinositol 4,5 bisphosphate, or PIP2. Without PIP2 bound to the ion channel, it doesn't function. So we thought, well, let's just see, maybe it's a loss of PIP2. And so just to give you a little background, PIP2 is a minority phospholipid that is an inner leaflet of the bilayer, and it's produced by PI4 kinase and PI5 kinase to produce PIP2. In this case, we bind and keep the ion channel functional. This requires ATP, and it's under dynamic control by GQ proteins, where if these are turned on, it chews up PIP2 to IP3 and diacylglycerol. So it's under dynamic control by synthesis and rapid degradation by turning on receptors that will chew up PIP2. So Osama then put PIP2 in the pipette, recorded currents from capillary endothelial cells from CADASIL, and lo and behold, putting PIP2 inside the cell had an immediate restoration of the normal current, and PIP2 by itself in wild type cells had no effect. This immediately suggested that this loss of current, this is the current from here to here, and from here to here, it's about 50% of the current is gone. We can immediately restore it by adding PIP2 back. So wow, this is pretty amazing. So Fabrice went to test this in the CADASIL artery. Here's the arteriole here, stimulates the capillary, and this shows the diameter of the arteriole. Look, capillary stimulation, no effect. Arteriolar stimulation, big effect. So the deficit's in the capillaries. Potassium's no longer turning it on. So we put PIP2, water-soluble PIP2 in the bath for 15 minutes, then repeat it. Amazing. We restored capillary signaling to the arteriole with 15 minutes of incubation with PIP2. So Fabrice restored this signaling that had been compromised, presumably for months in the animal, by 15-minute exposure to PIP2. So it looks like the cause of the deficit in blood flow in this model of small vessel disease is caused by a decrease in PIP2, which then silences about 50% of the inward rectifier potassium channels. I mean, this is just, I mean, actually Fabrice didn't want to tell me until we did a couple experiments because it was so mind-boggling. And I should say, I'll go back here, that this was a wild experiment because PIP2 works from the inside. So we weren't sure that it even do anything from the outside because it's an anion. And so we reasoned that all cell membranes have a lot of transporters, flipases, that move charged lipids from one side to the other side of the cell bilayer. So we assume it's being transported in. So we tested this in vivo by giving PIP2 IV to a mouse that has a deficit in functional hyperemia, a lower response and blood flow to sensory stimulation. So this shows the decrease in blood flow to whisker stimulation in CADASIL. PIP2 into the bloodstream of a CADASIL mouse restored blood flow response to whisker stimulation in 15 minutes, and it was barium sensitive. So this is really remarkable, been able to correct a deficit that I said these animals probably had human equivalent to for years with PIP2. Just one more sort of extending this a bit further, Amreen and Osama in the lab and looking at another model of impaired model that drives dementia. It's an Alzheimer's model, and all of these models are associated in humans with impaired cerebral blood flow. So they looked at a mouse called 5XFAD, which has 5 of the human mutations associated with familial Alzheimer's disease. Shows that they're 12 months old. They show amyloid pathology, plaque formation, and even at this stage, now neuronal cell death. And this shows control and shows all the plaques in this mouse model. And it shows that the mouse model, like CADASIL, the Alzheimer's mouse model, has this is whisker stimulation, blood flow goes up, and in the Alzheimer's mouse model, it's reduced by about 50%. And this reduction is the barium sensitive, inward rectifier sensitive component, just like in CADASIL, shown here. Barium reduces the control, but has no effect on the lowered blunted increase in blood flow in the Alzheimer's mouse model. It also, the Alzheimer's mouse model, capillaries no longer respond to potassium. So if you stimulate a control, the flux of red blood cells in the capillary to 10 millimolar potassium in vivo, well, in this case, about doubles. And in the Alzheimer's mouse model, there's no effect. So it's like CADASIL. This entire inward rectifier, retrograde electrical signaling, rapid increase in blood flow mechanism doesn't seem to be working. As in CADASIL, Osama found that was a dysfunction of the inward rectifier. The currents are reduced by, like CADASIL, by about 50%. And when he applied PIP2 externally, he restored within minutes the inward rectifier current, shown here and here. So again, PIP2 corrects it. Remarkable. And then looking through the literature, there's a large literature on altered phosphatidylinositol metabolism, PIP2 metabolism, and Alzheimer's. So this has, even though our experiments weren't based on what the other folks were looking at, there's also a line of thought that maybe what we're looking at underlies some of this pathology that other people have described in the past. So, remarkable, PIP2 corrects functional hyperemia, the increase in blood flow to neural activity in the Alzheimer's mouse model. Again, we applied IV, so it doesn't presumably cross the blood-brain barrier. It restored, here's the PIP2, look, the increase in blood flow goes back to normal. It's barium sensitive. So again, within about 15 to 20 minutes, restored the increase in blood flow to sensory activity, and it's barium sensitive. So remarkably, this seems to have a similar underlying reason for the deficit in delivery of blood to functional neurons, and it seems to be also corrected rapidly by PIP2 and likely involves the inward-rectifier potassium channel in the capillary endothelium. So impaired cerebral blood flow, impaired endothelial cell inward-rectifier activity, PIP2 corrects it and corrects cerebral blood flow. So here's sort of a summary of some slides. Normally PIP2 is required for the activity of this channel, the potassium sensor. When it's activated, it sends a signal upstream, causes smooth muscle relaxation and vasodilation. In Alzheimer's disease or small vessel disease, with CADASIL, the inward-rectifier potassium channels are there, same density, but they're impaired. You lose half the activity, and you lose the ability to send upstream rapid electrical signals that cause vasodilation, and you lose about half the increase in blood flow to sensory stimulation. If we give PIP2 back, we restore the control of blood flow and the delivery of blood during sensory stimulation in these two models for the neurovascular pathology, Alzheimer's disease model and CADASIL. So I'm about to end, and I want to show you a little bling at the end and show you there's another universe of signaling, not just electrical signaling. As you can imagine, if you think about your brain and the capillaries and all the wiring, that these electrical signals are going off all the time to signal upstream to tell arteries to dilate or then turn off to tell them not to dilate it. We also discovered that there's another set of signals that don't move, and there are calcium signals in the vascular endothelium. This is work done by Tom Longden, Amreen, and Grant Henning, and I would say all this work was, a lot of the work was done by Tom and Fabrice. This is more recent work, and this shows a vascular volume. The purple things are the blood vessels in the volume. It's 425 by 425 by 50 microns. You'll see the volume rotate around. There's a genetically encoded calcium sensor in only the endothelial cells, and then as it rotates around, you'll see it collapse into 2D, and then you see the third dimension will be time as the calcium signals emerge out of the capillaries and move upwards in time. And so you can see there's some enormous calcium signals out of the capillary endothelial cells and really little tiny ones. And we estimate in a volume about a microliter of brain has 5,000 of these calcium signals coming out of the endothelium per second. And other way of looking at each one of these calcium signals we think is caused by a local neural activity turning on the endothelium to release calcium. So each one of these splotches represents local neural activity, and we believe it's there to direct blood flow in a finer way, and the second way to with electrical signaling to make all this work. So one of the things we're trying to figure out is how does this all work together? How do all these signals merge in order to direct blood flow to all the active neurons and to sustain life? So lastly, some blue sky thoughts. And one of the things we're aiming for, the Holy Grail, would be, this is always search for the Holy Grail. Will restoration of normal cerebral blood flow control in small vessel disease improve cognitive function? $1 million question, or 10 million, or billion. We see that that PIP2 is a master regulator of cerebral blood flow and a linchpin that's controlled. All those calcium signals you saw are a result of IP3 going up in the endothelial cells. So what they represent, all those calcium signals I just showed you, they represent a place where PIP2 has gone down. Also, there's one discovery approaches to rapidly restore normal blood flow. So we've provided, at least through PIP2, control in small vessel disease. This has significant implications for restoration of cerebral blood flow following ischemic stroke. And then, so now for something completely different, the other function of local increases in blood flow is not just delivery of nutrients, oxygen, glucose, but it's also to get rid of waste, metabolic waste from active neurons. So perhaps we have it all wrong. What we're really looking at is really a waste removal process, regardless of its importance. So that's another aspect we hope to explore in the future. And now just some acknowledgements. This is our Woods Hole meeting. Tom, who's done a lot of the work on the original capillary sensing work. Dan Collier worked on histones. Albert Gonzalez just had a paper in PNAS and just took a job at the University of Nevada, Reno. Tom's at the University of Maryland as a tenure-track faculty member. Osama, who's done a lot of the work here and is now a research faculty, about to start on our COBRA. Dan Collier is a new research tenure-track faculty member at the University of Tennessee. And then here's another psychological thing. Can you tell by the organization of their lobsters? Too neat, creative, it's open to interpretation. Now, this is the view out our lab window in January, minus 20 degrees out. Fabrice, who did a lot of the work, is now a faculty member, as I mentioned, in Colorado. Here's our Leduc meeting. Fabrice is drinking red wine in Munich. Osama's been through beautiful work here. This is our lab outing with our Manchester group at Hadrian's Wall. It's a nice, beautiful February day. Here's the gang, our Leduc meeting, Albert, Masayo, Gros, and Fabrice drinking Guinness beer in Paris. David Healy-Banks, a longtime collaborator, great work. This is Susan Amar and Adam Greenstein, actually in South Burlington, now almost 2 years ago almost. Here's again the view of the lab, collaborates with Mike Kutlikoff. Here's Ann she got the brain prize with her colleagues from the Lundbeck Foundation in Copenhagen, May 9th, 2019. Here's the Crown Prince of Denmark. I forgot his name. He was a nice guy. Here's our lab. Masayo, Albert, Tom, funding group. Thanks again to the NIH. Acknowledge a few more people. Here's people in the lab, research technicians. Terry and Dan are great. Tom Hepner works on the bladder. Maria's doing great stuff on other aspects of the microcirculation of the brain. Longtime collaborators, Fernando Santana and John Lederer, chilling on Lake Champlain. Jerry Herrera comes back, was a graduate student 20 years ago and decided he finally got over me. He came back to the lab as a faculty member. And I want to thank everybody for having me. Thank you, and thanks, Susan. And thanks to lab. And I'm done.
Great. So you have actually a lot of questions, and hopefully some are really hard.
Okay.
I can tell you, okay, the first one is from Hakeem, who's a postdoc at Loma Linda University. So we have a pretty wide audience here. His question is, does the endothelial inward-rectifier potassium channel induced hyperpolarization move forward as well, like from arteries to downstream arterioles and capillaries? If so, in what condition may that happen? And he's interested in looking at it in Alzheimer's disease in his lab.
Well, that's a great question. The question about directionality, and this is one of the sort of our blue sky questions. The answer is I don't know. You seem to be, it could go bidirectionally, and how does it know when it hits branches? Does it goes both directions? The goal would be presumably to have the signal be channeled toward the upstream arteriole, but maybe there's a purpose for it to go towards the venules. I don't know, but I can say Nick and Aresh are collaborators into computational modeling. We had a paper in PNAS where they where they modeled this mechanism, and it becomes critically important, the number of active inward-rectifier potassium channels. So you can have two responses. K goes up, you get a robust hyperpolarization and a stable new negative membrane potential. Or if you get something in between, the membrane potential will oscillate and be unstable. And if you don't have that critical number of channels, it will fail to hyperpolarize. So it's possible that, and so how could it fail to hyperpolarize? Let's say all the cells have the same number of channels. Slight changes in PIP2 could cause that, as I showed in the talk. So each one of those calcium signals I showed in the last Bling movie from Grant and Tom and Amreen those represent GQ-PCR activity and capillary endothelial cells that have PIP2 being chewed up. So you can almost visualize those calcium signals as stop signs directing the electrical signaling too. So it's really, but ultimately we'll have to get some, which we're working on, to have a really good voltage indicator in the endothelium to look at signals simultaneously. So it's a great area. Great question. Thank you.
So from Mark Charette at NIH, does PIP2 restore cognitive function in the AD model?
Mark, that's a great question. Hey, welcome aboard. Don't know. We hope to test that, and that is one of the big questions. If we can correct this, initially in our mouse model, would we, do you have an immediate impact on cognitive function, and where there's a deficit already, or is it a longer term one? And I'll show you. Interesting enough, I have it on my desk over here, but not going to go away. But the lecithin, which is a over-the-counter supplement from egg whites, it's a bunch of phospholipids, including PI, phosphatidylinositol, and they market it as improving cognitive function. So maybe they have something right. But Mark, we're definitely planning to test that, and it's a really exciting area, as well as test which would be the best PIP2 derivatives to try. Or it's possible to try something downstream from PIP2, so great question.
Okay. Actually, your colleague Fabrice has written, wonderful talk, Mark. What would be the role of calcium and its possible impact on parasites?
I think it means calcium in the endothelium. Well, I didn't go into it, but all those endothelial cells express nitric oxide synthase, which is calcium activated, calcium calmodulin activated. So every one of those calcium signals, as Fabrice knows, would be presumably causing local release of nitric oxide. And we see when they're next to contractile mural cells, they cause a relaxation, local relaxation of the mural cells, which increases local red blood cell flux. And so that calcium signal that I showed, maybe it's a softball question, would presumably, and we have evidence for, would cause nitric oxide production to go up in that capillary endothelial cell and cause the adjacent pericyte to relax, and that would increase local red blood cell flux. But the longer term effects, there's also possibilities that the nitric oxide displaces oxygen from hemoglobin in the red blood cells, which would then increase oxygen availability. There could be also longer term effects on cell function through nitric oxide and cyclic GMP and protein kinase. There could also be effects on immune cells or microglia by the nitric oxide being released. So all stuff first, great stuff to do.
Okay. So Roland Owens at NIH asks, does improving blood flow affect blood pressure in your system?
We in vivo, we measure the blood pressure and heart rate, and the conditions we use, that doesn't change, and we try to do everything locally too. Some of the things, so we haven't seen any, but I don't know about PIP2 actually, to be honest with you.
Okay. we can move on. Another Cameron Clayton from Harvard. Great talk. You showed that neural activity does not trigger vasodilation when inwardly rectifying potassium channels are blocked. But how does blocking your KIR channel affect subsequent neural activity?
That's a really good question. I don't know, because we're setting up now to try to do with different wavelengths to look at neural activity and calcium and neuronal calcium and endothelial calcium and red blood cell flux simultaneously. But I guess the question is, if you don't have that delivery, what happens to the neurons in the area that's subserved by that penetrating arteriole? Now, there is still blood flow, but I guess the question is, when do you have too little, and how long, and what happens when you lose half of it over a long period? So that's a really interesting question. I know the concentrations of barium we used didn't affect, which we measured, neural activity. So that's a really good question. I don't know, but thanks for the question. We'll look at it tomorrow.
Okay. I was going to ask a question now that we've gone through the list there, and that was really what you showed with PIP2 was really interesting. But my understanding is CADASIL is somehow related to Notch3 and a whole series of events. Do the potassium channels, are there fewer of them? Is this just taking advantage of a mechanism for enhancing the activity of a limited number? Or is it actually somehow related to the etiology of the disease?
Yeah, so Susan, what you refer to, which Ann and I had a paper on, is the pathway appears to be Notch3, the mutant Notch3 in the extracellular domain. They oligomerize, they come together, and what they do is bring other extracellular matrix proteins into near the blood vessel, including TIMP3, which is a metalloprotease inhibitor. When the soluble TIMP3 increases, and that turns off, that inhibits atom 17, which prevents the shedding of HBEGF, which inhibits the activation EGF receptor. So we've done every step of the pathway. So we can turn down the inward rectifier and cause a pathology by giving exogenous TIMP3 in a mouse, or we can correct it by HBEGF. The pathway, so it looks like the EGF receptor is indirectly affecting the production of PIP2, and we think it's a combination of things that is leading, and Martin Dichon said some evidence, to a decrease in ATP, which decreases synthesis, but probably more realistic is both ATP, which we measured, is lower due to EGF receptor not being activated, and probably ADP is higher, so the phosphorylation potential changes. So we can restore everything by giving ATP, increasing ATP in the pipette. So it is linked to the pathway. It was kind of unexpected because it's, but we can mimic everything by manipulating that pathway as well. I tried to present it once and I almost killed myself because there are too many players. But if people are interested, look at the paper we had in eLife a couple years ago. We did everything, but we didn't link up the functional hyperemia to the extracellular matrix pathway. And this is a linkage.
Okay. So one more question came up from Dane Liston. Do gamma secretase inhibitors have an effect on cerebral blood flow due to Notch3 reduction?
Good question. I don't know, so that's a simple answer.
Okay. So I think we've reached the end of our hour, and I just want to thank you for giving a really exciting talk. I didn't read all the compliments that you also got. So anyway, it was a pleasure having you, and it was a really fascinating talk, and I think it appealed to lots, and I'm getting a lot of great comments coming in.
I just tell you, it's fun work, but it's also fun having great people in the lab and great collaborators and seeing them being successful.
Yeah, well, they all seem to be, so that's great. So I think I guess we'll sign off. I'm not sure how we do this, but
Do I
thank
stay on here?
No, we have another Zoom
event.
call for you.
Okay, thanks, Chris. Hey, thank Chris and Karen. Thanks for everything. And
And-
Susan, I hope to see you in person sometime next year.
Yeah. And thanks to everyone who attended. So
Yeah,
it's
thank
great.
you, everybody. How many people attended?
Several hundred, I noticed that online, over 200.
All right, cool.
Great.
All right, thanks, Chris. Thanks, Susan.
Okay,
Yep.
so have fun. You're going to meet some of, I think even some of my folks. So.
Okay, cool. All right.